I decided not to bug you for your CV. So.
Oh, I sent the bio to Brian.
Okay, it is my pleasure to introduce Dr. Karen Holl. She did her undergraduate at Stanford University, a PhD at Virginia Tech, was a DOE Distinguished Global Change Postdoctoral Fellow at Stanford University, and then I believe after that ended up at UC Santa Cruz in the Department of Environmental Studies, where she has been ever since. She is best known for her work in tropical ecology and restoration ecology. You could even say she wrote the book. She at least wrote a book on restoration ecology a few years ago. And so she has done this work, a lot of her work in, or most of it perhaps, in Costa Rica and elsewhere in the neotropics, and also has done this work in a diversity of terrestrial systems in Central California. And so in restoration, we often think and talk about the importance of landscape context and landscape processes. And so this is something that she studies and has incorporated into her work extensively, some of which I think we'll be seeing today. And then something just in general that she does enviably well is that she does basic science and testing of theory, but in the context of like meaningful applied questions. And so she has been recognized by the ecological community as a fellow of the Ecological Society of America and also the California Academy of Sciences, but then for her contributions to practice and policy, also an Aldo Leopold Fellow, and then also an award from the Society for Ecological Restoration. So I am very much looking forward to this talk, and I'm sure you guys will enjoy it.
Thanks. So yeah, it's great. Thanks. It's great to be here today, and thanks for everybody who's hosted me. I've had a lot of really interesting conversations. I've been to Utah, but this is my first time in Logan. It's beautiful. A little colder than California, but that's good. So I will be talking about my long-term project in Costa Rica today. It is very much a we project, a lot of people involved in it, some exemplary grad students. My co-PI on the project is Zach Zahawi, who's the director of the Charles Darwin Foundation in the Galapagos now, and many Costa Rican field assistants that have been instrumental to the project. But before I do talk, I did want to say, I will be talking about this project today. I do work broadly in the tropics. I have collaborations a little bit you'll hear about today and then tomorrow in Brazil. I've worked with students in Colombia and Mexico in the tropics, mostly working besides this project very much with collaborators in country. But I do have a research program in California, do some large scale research on riparian forest restoration along the Sacramento River, and work locally in the Santa Cruz area on grassland restoration, particularly with a focus on some of the rare wildflower species. And I know that's been I'm happy to talk to people about that too. And I have already talked to people about today, but if you have questions. And then as Kari mentioned, one of the things I'm proud of is my primer of ecological restoration that came out a couple of years ago, which provides sort of a succinct overview of the field. And I'm pretty excited because hopefully this year it's going to be coming out in Spanish, Portuguese, Chinese, and Farsi. And I'm really excited, particularly since I work in Latin America, to make it available to the people I work with there. So with that, I'll move on to my, okay, clicker is not working for reasons I do not know. There we go. Okay. So there are many forest restoration and tree planting campaigns globally. For instance, the Bond Challenge commits to or has a target of 350 million hectares by 2030. That's the area the size of India. There are at least 3 global trillion tree planting campaigns, and I'll be talking more about that in my talk tomorrow. We're now in the UN decade of ecosystem restoration. But the big question is, why the clicker is not working. I'll stand over here and click. Can I click manually or no? Oh, maybe, okay. But the challenge is scaling up space and time. How are we going to restore at this space over such large scales with limited resources? Oh, that does work better. So one way to restore is to natural regeneration is one option to regrow forests, but the rates of forest recovery vary greatly. And so in some areas, it can be a really promising approach. For instance, this is in the Yucatan Peninsula, 2 years after shifting agriculture, huge amounts of regeneration. in contrast, this is another site in the canal zone in Panama. And what we see there is that after 10 years, there's very little change because of these aggressive grasses. So that's a place where we'd have to intervene. So one of the things that we really think about is, what explains the differences in these rates? And that requires an understanding of the ecology of the system. And so we know that in these systems that have been used for agriculture for a long time, first of all, the seeds have to arrive, they have to survive, then they have to germinate, the seedlings survive and grow, and then so the trees need to reproduce to maturity. So we have all these life history stages, and those are affected by a lot of different factors. And so one of the things I've done in my research and working in the tropics for about 25 years is really try to understand which of these factors are most important. So we have to look at things like, brave this pointer here. I'm told if oops, that's the wrong one here. Okay, there we go. Look at I got it to work. Okay, so like in the tropics, where about 80% of the mature forest trees are dispersed by birds and bats, so we need to seed dispersal is often limiting. But you can have a whole other host of factors like seed predation and herbivory, microclimate, pathogens, soil physical and chemical properties that all affect these transitional stages. And in a system where you have up to 500 tree species, you're not going to be able to go in and replant all those trees. So I think it's really key to understand the ecological processes, figure out where those impediments are, and then design your strategies to overcome those barriers. Now I am pointing. Oops, there we go. Okay, whoops, shoot, now I got it back. Okay. So one of the ways to overcome those barriers and the most widely used strategies is to plant native trees, which that overcomes a few different barriers. It helps to enhance seed dispersal by birds and bats. It shades out pasture grasses and moderates microclimatic conditions. It also can, if you plant nitrogen fixing species, it can increase nutrient availability. But the problem is that it creates really homogeneous conditions when you plant rows and rows of trees, and it's also costly to plant and maintain large numbers of seedlings. So this got me and my co-PI thinking as ecologists, how might we do this in a better way? And so this brought up the idea of the nucleation model of forest recovery. And one thing that we know is that forests don't recover in rows and rows, they recover patchily. And there was this article about 50 years ago that looked at old fields in the temperate zone in the United States and realized that they tend to recover by nuclei, and then they spread over time. And you can see that in post-fire recovery. You can see that in a lot of different systems. And so in the tropics, the potential would be here that you have these, come in, they disperse, whoops, pointer down. You attract certain dispersers, then over time, these nuclei grow. And even though this was originally proposed in the temperate zone, we see this sort of process happening in the tropics. So what we did was to think about this. If we know that natural regeneration, in some cases, it's a really good strategy for recovery, but it can also be quite slow. We know that plantations are homogeneous. So then the idea would be, let's go out and let's plant these sort of patches of trees, and then over time, they will grow and spread. And so this was sort of our concept. And we set up the study. And I asked the questions of, first of all, does it facilitate recovery? What is there a critical nuclei size? And I will use the term nuclei, and we call them tree islands too. So is there a critical size of these tree islands that we plant? Do the nuclei spread over time? And also just thinking about, from a practical standpoint, how practical is it to implement this type of an approach? Okay. The other question that we asked in my work was this question of local versus landscape scale recovery effects. And so kind of the question is it more important where we restore or how we restore if we're going into restore? So to do this, what we did was we set up a large number of different sites across the landscape. Some of those sites are, I don't know why this isn't working. Okay. So some of those sites are surrounded by a large forest cover. The forest cover is green. Other ones are out in the middle of the agricultural landscape. And so we also were looking at this effect of the surrounding landscape on recovery, as well as the within site treatments. So the area where we work is in southern Costa Rica. Actually use the pointer on here. I guess I put the pointer here. It's a fragmented pre-montane forest. It was used for a long time for a lot of coffee, but in recent years, a lot of it's transitioned to cattle grazing. A couple of people today have asked me where it is. We work here in southern Costa Rica. Literally, we can walk to the Panamanian border, so it's not where most people have visited if you've been to Costa Rica. We did intentionally set up a large number of sites, and you can see they're spread across the landscape. And then the sites that have the dark black circles around them, those also had reference forests next to them too. And we do get quite a bit of rainfall every year, and it's pre-montane forest, so it's at about 1,000 to 1,400 meter elevation, so it's not sort of lowland tropics. But you're right, maybe I'll just have you just say next slide. Okay, sorry. Okay, so we originally established 16 sites. The idea was to establish a number of different sites. That was in 2004 to 2006. At this point, we have 11. When you're doing an applied study across the landscape over time, we have lost a few sites due to different reasons. We separated by the sites by at least 1 kilometer. And then in each site, we have 3 different treatments. The first treatment is just natural regeneration. There's no planting. Then we have plantation, where we planted the entire area. And then we have 3 different nuclei sizes, 4 by 4, 8 by 8, and 12 by 12. We planted 4 species of trees. 2 of them are more timber valuable tree species. And then these 2 lower species are nitrogen fixers that are commonly planted in coffee plantations. And that was partly based on what we could get at the time. We planted them 2.8 meters apart. And then we also cleared the grasses for 2.5 years. And since that time, we've just let it go. I'm pretty proud of the fact that this is, I would say, probably the longest running, best replicated tropical forest study that I know of, that we've actually been able to really look at the forest recovery over time. Next slide. So just to give you an idea what the site looks like, this is one of our sites, and this is at after 1 year. You can see the person, you can see the little seedlings. Here is the same site at 9 years later. So there's the person and there's the canopy. And so it's very different than working in the Western United States, where things grow a lot more slowly. In the foreground here, we have, this is one of our natural regeneration plots. And you can see that even after 8 years, there wasn't a lot of natural regeneration in this particular plot. Next slide. So this is just gives you an idea of what the sites look like. The plantations, the canopy closed within about 2.5 years. This is an artist rendering, a student who worked with me, but you sort of emphasize that they were planted in lines. There's some natural regeneration under it, and there has been some mortality of trees, so it has opened up a little bit over time. Next slide. The natural regeneration, the sites vary quite a bit. And so in some of them, there is more, but a lot of them, they're still, even after 18 years, covered by dense grasses. And I like to point out that these aren't like grasses that you're used to around here. These are grasses that, this is the person, this is the grass. So at some of our sites, the grasses are about 1.5 meters, and other sites, they're up to 3 meters. And so this is, you can imagine if you're a seedling trying to establish under those grasses, that's rather challenging. Next slide. In the island treatments, this is an island after about 2.5 years, and you see sort of these individual islands of trees. Next slide. But what happens over time is the canopies start to merge, and so this sort of shows the edge of the canopies merging. And then here in the center, we have other tree seedlings that are recruiting. Next slide. I also like to point out one of the things I think in any ecological study, but particularly in restoration, it's really important to replicate at multiple sites. So I sort of showed you photos of what my characteristic sites look like. We also have a couple of plantations that look like this, more like a savanna. It didn't grow quite as well. And one of the things that we find is a very strong variation in the growth rates, even in this one region across sites. And I figure that any trend we can actually see amongst the noise of our data, we can really conclude fairly well, because we are working across the landslide. Next slide. And I think it's really important to have multi-site experiments. It also makes me a bit concerned about how much carbon we're promising to sequester in different sites, since I see like an order of magnitude different in how much carbon was sequestered in the planted trees at my sites. Next slide. We spent a lot of time scratching our head about what explains the differences in the variability. And we did a lot of soil nutrient measurements. We've measured infiltration, compaction, all sorts of different things. We have done work on mycorrhizae. Never mechanistically could we explain the variation very well, but the one thing that was really interesting is we did take site histories, and we found out that the length of time that the land was used for pasture explained 2/3 of the variation in the tree growth rate. And in an ecological study, if you can explain 2/3 of the variation, that's pretty good. And so I don't know exactly what it is about the cows, probably some combination of different factors, but definitely the longer the land is grazed, the slower the recovery rate. Next slide. So over the last 18 years, we've collected a lot of data on a lot of things, planted tree growth and survival, vegetation structure, seed rain, epiphytes, birds, bats, insects, lots of different stuff. There's we're up to about 60 papers at our website. Don't worry, I'm not going to tell you about it all today. What I'd like to do is sort of emphasize some of the main take-home points. Next slide. And I think some of the grad students read this paper, but this is a good sort of summary paper of a lot of the results that we learned. And so I'll talk about a few of those lessons today and a few highlights, and then also talk about where we're headed with the research. Next slide. So lesson number one that we found and we feel pretty good about is that it seems like applied nucleation is similarly effective to plantation treatments in the recovery of most variables. And I'll just show a couple. So here on the y-axis, we have, this is seeds per meter squared per year. So this is, these are small seeded species. And then these are larger seeded species down here, so greater than 5 millimeters. And most of the later successional species in this forest are larger seeded species. And so what we see is that if we look at seed rain, that at first, in the very first few years, this is after about 6 to 9 years in our sites, there was a lot more seed rain of all types of species, but over time, the smaller seeded species don't seem to be that dispersal limited. We have a lot of variability, but there are small seeded species getting to our sites. However, if we look at the larger seeded species, we see very clearly that applied nucleation and plantations have many more than natural regenerations, but those are still a lot less than the reference forest, probably because in the reference forest, a lot of the seeds are dropping directly from the plants, which they aren't in the restoration sites. Next slide. If you, this on these right hand show the same figure, but it shows the number of tree recruits per meter squared, and you see a fairly similar pattern. We see that for smaller seeded species, they're lower in natural regeneration, and they're actually similar in the applied nucleation plantation and reference forest. For the larger seeded species, we see the same exact trend as for the seeds, that you have a lot less than in the reference forest, but that applied nucleation and plantation are very similar. Next slide. So this is a newer paper that just came out this year, and this is looking at the trends over time. And what this shows is again, these are the naturally recruiting seedlings, not the ones we planted. This shows overall species richness on the left figure here. And then this is functional richness on the right figure. And you see the same trend, which is that over time in the blue and the green, the applied nucleation and plantation are going up. It stays relatively level in the natural regeneration, but it's still well below the reference forest. And so that they are recovering, but we still have a long way to go to get towards the forest, and particularly with respect to species richness and composition. Next slide. This figure actually shows bird data. We have a long-term bird data set, and my graduate student, Francis Joyce, is in the process of analyzing it. What we see here is a fairly similar trend. This shows bird community similarity, the community similarity to the forest. And one thing he did was look at the similarity across the reference forest, which I think a lot of people don't look at. And even across the reference sites, similarity is only about 35 to 40%. We do see that over time, the plantation and the applied nucleation are showing a similar trend as the as with the plants, but they're actually getting closer to the reference forest. We also see a little bit more recovery in the natural regeneration plots with respect to the birds. And then we've also been sampling in reference pastures where we don't see recovery. So again, they're getting closer to the reference forest, but there's still differences. Next slide. So another lesson is we're finding that things change really quickly over time. You could see that in along those trends. But one example of something that really changed quite rapidly between about 5 years and then 12 years was looking at some of the nutrient cycling that was going on. So we've done leaf litter sampling and looked at the leaf litter nutrients that are coming into the plots. And I had a master's student who did that at year 5, and then another one that did it about 10 to 12 years after that. And in the second sampling period, we also sampled reference forests. Next slide. And I'll sort of walk you through this figure. So we've got leaf litter biomass on the y-axis here, and then we have the first sampling and the second sampling in each of the different treatments. So what we see in this first slide, again, the letters show significant differences. As one might expect, after 5 years, there was lowest leaf litter biomass falling in the natural regeneration, intermediate and applied nucleation, and highest in plantation. Next slide. But by 10 to 12 years after recovery, what we find is that we actually have that the applied nucleation and plantation have actually achieved similar levels to the reference forest over time. So they've actually recovered. It's still quite a ways below in natural regeneration. But what's also really interesting is what's happening to the composition of the leaf litter. Next slide. What we find is that in the plantation treatment, the vast majority in the early years of the overall canopy cover and associated leaf litter biomass was dominated by a single species, Inga edulis, which is a nitrogen fixture. And so we had really high nutrient inputs into the plantation treatment. And we were kind of worried, like we're completely changing the nutrient cycling because we've chosen a nitrogen fixer there. But what we found by the second time, or our second sampling, was that had dropped considerably. And the reason for that is we've actually over time had quite a bit of mortality of those species. So at the time of the sampling, we had about 50% mortality. As of the time this past summer, we had only 25%. So these species have just died over time, which in some ways is a really ideal nurse tree for restoration because it shaded out the grasses and now they're sort of allowing the other species to come in. Next slide. So what happened was that as those species died, what we found in the planting treatments is that one of our other planted species, Vachisia guatemalensis, took over and dominated very much the plots. And so it sort of compensated for the death of the Inga. Next slide. But what happened in the natural regeneration and to a certain degree in the applied nucleation is that you have other species in blue here that are starting to dominate the biomass. And so you have this you have a lot more species coming in. So that's another argument for why you might want to use an applied nucleation approach, because you're not, this really dense canopy that's shading everything else out. Next slide. Another question we asked at the very beginning was what the most appropriate island size or nuclei size was. And as a reminder, our large islands were 12 by 12 meters, the intermediate were 8 by 8, and the small were 4 by 4. And then we measured both seed rain and tree recruits at different distances going out from the edge of the planted area. The trend for both the seeds and the tree recruits is pretty clear. Large and medium islands result in a lot more seeds and recruits, and the smaller islands were not particularly effective in elevating the amount of recruit establishment compared to just being outside. Next slide. So we think there are probably two mechanisms for that. First of all, the smallest tree islands did not attract as many seed dispersing birds. The other thing is with those smaller islands, if like one tree dies, then you have an open area and there's only 5 seedlings in there. There's 4 at the corners and 1 in the center. They don't shade out the grasses as well. So it seems like in our system, and this would obviously vary with system, that there's sort of this critical island size of about 10 by 10 meters, that you need to have to be able to shade out the grasses and for them to establish. Next slide. The other thing that we found very clearly was, and this is key to the nucleation strategy, is that the tree nuclei have spread over time. This shows an aerial photograph from about 2 years ago, and you can see the plantation is very dense. The applied nucleation, the islands have merged quite a bit, which is why I don't have later data on island size, because they've already merged with one another. And it's still pretty patchy, but there is some taller canopy in the natural regeneration. And as I showed previously, they're starting to merge in a lot of the sites. I like to point out that this is true in our system, but this is not true in all cases of using this approach. Next slide. This is an example from Spain from a colleague of mine, Jose Maria Rebanayas, and he planted oak islands 21 years before this photo, and you can see exactly where he planted the oak islands. And these are the only these little green dots are all the recruits over 21 years, and you can see that they really haven't expanded. And so when I was out there, when I was visiting him on sabbatical, we noticed that like there's tons of sort of the grasses sort of lower along the edge. Next slide. And so he did some experiments, and sure enough, there's like increased herbivory along the edge. And I actually find that in the coastal sage scrub in California where I work too, which is that you tend to get a lot of rodents and things that sort of are inside, and then they come out just to the edge to eat. And so there's a really high herbivory pressure there. And so in that particular case, probably an applied nucleation strategy wouldn't work as well. Next slide. The other thing that we found is that, so applied nucleation is cheaper than plantations because you're planting much less area. So in our case, we planted about like just over a quarter the amount of trees. So obviously there's certain fixed costs like land, but the cost of planting and maintaining the trees is going to be much less if you're not planting as much of the area. So that's a pro, but next slide. We found that there are some logistical and social challenges to using applied nucleation. Next slide. First of all, when you put the seedlings out in these islands, it's much, and then we had people would come up and they would like clear around the seedlings, either with a weed whacker or machete. It's much harder to know exactly where your seedlings are amongst the grass if they're planted irregularly, as opposed to in a line. So there was slightly more damage to our seedlings from just accidental macheting. Next slide. But the bigger issue that we found, and I talked to my colleagues who work in agroecology about this too, is that a lot of landowners do not like messy landscapes. We as ecologists say, hey, heterogeneity, it's great, but there's this whole sort of landscape architecture view that things need to be sort of well kept. The other thing is that there's also issues with like land in an agricultural system like where I work. Like you need to show that your land is productive. And if people are growing trees, then that is productive. And this is looked at as messy. And so we had problems with like people trying to put cows on our plots during the early years because there's grass and it's not being used, or like, why do they look so messy? So I think that's something else we really have to think about is social preferences. Next slide. So as I mentioned at the outset, another question we asked was about landscape effects. And so how much surrounding forest cover affected recovery. Next slide. I haven't talked as much about that. And that's partly because in the first about 15 years or so, we have not seen huge surrounding forest cover effects, which is sort of surprising, and it goes against what a lot of landscape ecology would tell you. I think we have seen some effects. We have found that the amount of surrounding forest, for instance, affects the composition of birds. So you tend to get more forest-dependent birds if you have more forest surrounding the site. We also had more species richness of angiosperm epiphytes, but for the most part have not seen a strong forest cover effect. So one reason I think that is because in this particular landscape, there are a lot of tree elements in the landscape. So you have things like these living fences here, when it shows up, these living fences here, that trees along here, you have isolated pasture trees. You also have riparian sort of forest fragments in there. So there are enough trees in the landscape that there is some movement and sources of seeds to colonize. And so I think that's one reason. There's areas where I work in Brazil where there's almost no trees, and then you find a lot more of a landscape effect. The other reason, next slide, is that this is just doing analysis of forest cover. And one of the things we started to realize was we would get an oak, and the seeds of oaks in the tropics are quite large, and it would be out in one of our very low forest cover landscape, and it'd be like, wow, how did an oak seed get this far? And our Costa Rican project manager would be like, and there is the mother tree over there. Now, we don't know that this oak over there was the mother tree, but given that, because we didn't do genetics, but given that there's like one oak tree in the landscape, it's like, that's probably where it came from. And so we started looking at actually mapping individual tree species. And so we went out and mapped, we did map the trees, potential tree species within about 100 meter squared 100 meter radius around it, and then looked at, did distance to actual seed source have an effect? Next slide. And sure enough, there was a strong effect of the distance to mother trees, particularly for large seeded animal dispersed species. And we're continuing this work in our new stuff, in our new project, and we're using more remote sensing data to try to be able to look at it even a larger scale. But what it suggests, which is not surprising, is next slide, is that it's not just the amount of forest cover, but it's the, next slide, it's the composition of the remnant forest is really important. And in our case, there are often seed sources within the agricultural landscape, which advocates the importance of how you manage your agricultural landscapes. Next slide. So in conclusion, applied nucleation is a cost-effective restoration strategy that simulates natural recovery. Next slide. Larger islands have several advantages. They're just easier to plant and maintain, and they also are much more effective. Next slide. Proximity to propagules of forest species is important, particularly as succession proceeds. And I think we're going to see more of an effect as we have more later successional species establishing. So it's the quality, not just the quantity of forests. Next slide. And I really think that applied nucleation should be tested, used more widely when it's consistent with restoration goals. And because we have to really think about which landscapes, not just where does it work ecologically, but where does it work socially. Next slide. So our ongoing research, we now have 18 years of data, which is pretty rare in restoration experiments. We're continuing to measure tree growth, vegetation recovery, and birds. And what we're really focusing on now is recruitment of later successional species and really looking at, what is limiting this establishment of later successional species, which most restoration experiments can't do unless they're using a chronosequence of sites, but we have data from the outset. Next slide. So this is our conceptual proposal from our recent NSF grant. And what we're looking at is, so what we're looking at is both the later successional species and looking at small seeded versus large seeded species, and sort of, as one would expect, that distance to source is going to have a big effect, particularly for the larger seeded species, but that seed dispersal is probably very limiting for the large seeded species, but establishment limitation not as much. And then actually that growth will probably be higher, even though we know that dispersal is lower in the natural regeneration. We've actually found some evidence that growth is higher because there's more light, and that ultimately our hypothesis is that you'll get obviously the highest will be in the reference forest, but that in the applied nucleation, so the combination of being able to have more open light situations, but also having trees, those will actually result in higher diversity and similarity to reference forest, but we'll see. So next slide. Okay. So what have we been doing? We have about 500 seed traps out that we are collecting seed rain on for 2 years. We're at about 1.5 years now. And it gives me an opportunity to point out the most important person on the project, which is Juanabel Rosales, our Costa Rican manager, who has been working on managing the project for 18 years and does a lot of our seed rain ID, plant ID, now he does bats and birds. He knows way more than I do, much more important on the project than I am. And we're also doing various seed addition experiments. This is my graduate student, Frances Joyce, who's been doing a bunch of seed addition experiments, some with herbivore exclosures, some not. And then the other thing that we did is we actually, normally in a given year, we sample subplots within our plot for recruits. But what I've really been wanting to do over the years is to really do a complete thorough survey of our plots. So this last summer, we went out and we did a thorough recruit. We surveyed all of the seedlings greater than 20 centimeters. Next slide. And we ended up with about 84,000 seedlings of about 260 species. This photo is exemplary of the summer, which is that it rained twice normal. So it was really fun trying to get out and sample all these in the middle of it. I was fortunate to have a really good crew. We also had like some wasp outbreaks and COVID outbreaks, but we made it through. And so we're just finishing putting together all the data sets and sorting out all the kind of irregularities in the data, we'll call them, but really excited to analyze this data set. And this will allow us to look at some questions of heterogeneity in the plots and just really have the sample size to be able to ask some questions we weren't able to ask in the past, but I don't have the results yet. Next slide. Some results I do have from the seed predation germination experiment. This is a slide from Frances's work, which is in progress, but this looks at the percent seedlings emerging of 2 of the large seeded species, where he did do large herbivore exclosures. And he's also been doing a lot of camera trapping, and he's actually found about 60 different species of mammals and birds in his camera traps, not all of those interacting with the seeds, but we've had more bird seed predators than we had anticipated. You can see really clearly that obviously predation is a huge factor, because in the cage treatments, there's a lot more seedlings germinating. Not seeing a really strong difference between the plantations and the reference forest. And generally, we aren't seeing these. We only did exclosures with the large seeded species because the smaller seeded species were just too hard to re-find. But thus far, germination is similar for most species across all treatments, both large and small seeded. And so it seems like it's probably more predation and dispersal limitation of these larger species than it is germination per se. Next slide. So the other thing that we're doing is we are also trying to, I think it's really important to test, as I said, to test this approach at other sites. And so we've been involved in different projects. I'm going to talk about one in Brazil that I'm involved in Zach, my co-PI, has been working with some groups in Colombia, and one of my former grad students, Leighton Reed, has set up a new big project in Ecuador, looking at applied nucleation and specifically looking at distance between the nuclei, which is something that we did not manipulate. And I think by testing this in different systems, it'll give us a better sense of like when it is and when it isn't appropriate, and sort of where it works, and testing different questions. Next slide. So I set up this project with my collaborator, Pedro Brancaleon, who's at the University of Sao Paulo. And so he set up this experiment with nuclei. The green are planted and the white is not. So nuclei at 25% and 50%. He made the case that he didn't really think nuclei were going to be very effective in their system and it would make a lot more sense to plant in strips. And the reason he wanted to use strips was because, first of all, in Brazil, it's flatter land than where I work, and they are planting in rows, and they do it mechanically. And so it makes a lot more sense just to put the rows in and plant. The other thing that they do is they also subsoil. And so if they plant this way, they have to subsoil the entire plot. Whereas in this system, they just run down and they subsoil the strips and do it that way. The other thing was exciting because one of the things about doing long-term research is you make certain decisions right at the outset, and then you realize you should have made a different decision, and you have to go look at those mistakes every year when you go out there, and you say, like, why did I select those species, or why did I do it this way? So I just say that to the grad students. We all ask those questions of ourselves. One of the things that I would do differently if I started my Costa Rica project now is I would not have only used 4 species, of which 3 are wind dispersed. So when we reset this new project up in Brazil, they have very good planting schemes and species selections. And so half of them are animal dispersed and half are wind dispersed. And so and some of them are slower or faster growing. So it was a much better species selection choice. Next slide. So this is just sort of shows some overhead of it, and you can kind of see in the strips where you have the rows, and then this is using aerial photograph and looking at the canopy height here. I had hoped we'd have more data, but I was supposed to go there on sabbatical a year and a half ago and didn't get there for obvious reasons with COVID. So the data collection got a little bit delayed. Pedro did finally collect a really thorough round of data. It's now about 5 years old. This past April through June. And so he's in the process of processing the data, and I'm looking forward very much to him sending that to me. Next slide. What I did see when I was able to visit this April and May was that what they're finding in their system, and this is semi-deciduous forest, and my camera wasn't working very well that day, but what they're finding is that it is working in their system, but it's not shading out the grasses as fast. And that's because they get about 1,500 millimeters of rainfall, which is about half what we get. And the trees are growing more slowly, so they're not shading out the grasses as much. They also use a wider diversity of species, and so some of them are doing better than others. One thing they are finding is that they do think the strip planting is much more effective, and not just for the practical implementation reasons, but also because since it is drier, there's less edge effects if you have these long strips than having all these islands. And so it seems like just like particularly during the dry season, that it works better having the strips. So I'm hoping to get down there a year from now, and we're going to do even more data collection. So hopefully there's no more pandemic. Next slide. The other interesting thing that Pedro's been doing, he's really looking at thinking about how we could implement at scale. And so one of the things that he's done is looked at using eucalyptus as a nurse crop, because Brazil is the largest pulp paper producer in the world. And so what he's been doing is this experiment where you plant like 4 rows of eucalyptus, and then you plant 4 rows of native species. So rather than just leaving that intermediate strip to just have to manage it or clear it out, is you do that. Next slide. So then what it looks like is the eucalyptus grows really fast like this, and then you log out the eucalyptus after about 5 to 7 years. Next slide. And it looks something like this. And so by doing this, you help to offset your restoration costs. And he's been working with a number of the paper pulp producing companies. And I think it's one of the ways to kind of think about practically how we're going to scale something up like this. And I forgot to say on the last slide, earlier, a few slides ago, is that I've sort of, even after though I spent like 15 years using this term applied nucleation, I've started to think more about the idea of thinking about calling it spatially patterned planting is I think there's a lot of different ways we can plant and think about it more creatively than just planting rows and rows and trees. And sometimes it may be just choosing on a landscape where you plant. But the idea here is that these native species will grow up over time and then they will facilitate the establishment in the strips in between. Next slide. So continuing on the scaling up, the other thing that I've been involved with is trying to work with some of the NGOs to try to get this implemented at a larger scale. So I've been working with Conservation International, and a year ago we came out with this guide for applied nucleation in tropical forests. And I've been working, doing some advising on a project in Rondonia where they're starting to try to use this in about a 500-hectare plot. And so trying to think about how we could actually move this, like I said, to scale. Next slide. So in closing, I do think that, you know, applied nucleation could be a useful restoration strategy. But like any restoration strategy, you have to look at the ecology of the system. You need to look at the social system and think about where different strategies are work. And so we have this different toolbox for restoration managers and saying, okay, what works in this particular situation? I think that the places that these types of spatially patterned methods are most useful are probably in areas where natural regeneration occurs slowly, because it occurs quickly, then you really don't need to do anything. Or maybe you just introduce some of the later successional species that are slow to establish. In large areas to restore with limited resources. In areas where the spread of the tree nuclei or strips is not inhibited by things like herbivory, invasive species, or fire. I have done some work in California grasslands in trying to use nucleation plantings. And what I found was that because California grasslands are dominated by invasive species, rhizomatous species will spread because they can spread out, but it's very hard to get the other species that established by seed to spread because they just can't establish within the grasslands. And of course, you also have to think about whether it's going to be compatible with landowner preferences. And probably in small land holdings in agricultural lands in the tropics, this may not be the best approach. But if you're working in like larger areas like this, where CI is working with landowners and they're taking over sort of former plantations and things like that, this is the sort of type of area where it would be potentially more compatible. Next slide. So again, thank you very much to a ton of people who have worked on this project, and to various funding sources, in particular NSF, which has funded 4 rounds of this project, which is pretty unusual because they usually like to give things to new projects. And in particular, like I said, some of my Costa Rican field assistants without whom this project would not continue. Thanks. And I would be happy to answer any questions. Oops, I'll just turn this off, or what we'll do. I guess we can just go just turn it off. You don't need to see my screen here, but anyways, we'll go ahead.
So I was curious, you referred to this briefly, but early on, what were your objectives that you were shooting for? I mean, there are a variety of things you could come up with: carbon sequestration, wildlife habitat. And it seems like those really would drive how you want to do it, as you hinted on at the end. But, you know, I was looking at your pictures of the plantations, which seemed very corn-like, you know, like a monoculture. And yet you were ranking them as being very close to the reference